Hi everyone, welcome to today's webinar, Nano 101, a review of the art and science of nanotechnology. We're glad you could join us. My name's James, I run webinars and events here at Triumvirate Environmental, and I'll be your moderator for today's event. Before we get started, I'd like to give you a brief overview on what the webinar is going to entail as we run through just a few housekeeping items. You may have noticed when you signed on that your microphones are muted. We ask that if you have a question for our speakers or a technical issue, to use the questions box on the right-hand side of your screen, and I'll be able to respond to those. Following our presentation today, there will be an open question and answer period. The goal is to save about 10 minutes or so at the end to get to those questions, so get them in as quickly as you can. And if we don't get to them all, we'll be able to email you after the webinar and provide some follow-up. Also, we'll be announcing an offer after today's webinar, so be sure to stick around for that. And keep in mind you will get a copy of the recording and slide deck tomorrow. Our speakers today are Mark Liffers, Practice Director of EHS Consulting at Triumvirate, where he provides on-site technical support and outsourced program management to a wide range of technology-based manufacturing and construction clients. Areas of technical expertise include biosafety, industrial hygiene, construction safety, and others. Mark has a Master of Science degree in Industrial Hygiene from the Harvard School of Public Health. Dr. Jason Crescioni is the owner and Managing Director of JM Crescioni Consulting, a small independent scientific consulting firm that provides expertise in biomedical engineering and materials science and services for guiding the formulation, pharmaceutical development, and clinical translation of microparticle-based and hydrogel-based drug delivery systems. Prior to joining JM Crescioni Consulting, Jason was the co-founder and CTO of an early-stage specialty pharmaceutical company focused on developing drug delivery systems for the treatment of neurological diseases. Dr. Crescioni received his PhD in biomedical engineering from Yale University in 2012. And so with that, I'll hand it over to you, Jason.
Thanks, James. So today, what this webinar is going to focus on is a high-level overview of nanoparticles, the types, kinds, sizes, shapes, and geometries, as well as fabrication methods and ways that these types of materials are handled and processed in different laboratory environments. And then it'll go along, I'll toss it over to Mark. Mark will go through some of the industrial hygiene issues around handling and managing these types of nanomaterials in the laboratory environments. Before we get into that, we'd like to ask a poll question of the audience that's currently on the screen, which best describes your facility. And if you can answer that question, that would be great.
We'll give everyone a few more seconds here and share the results. All right, looks good. Get a couple more in. Okay, let's share those. So Jason, we've got 65% of our audience is academic, 21% R&D, 14% industrial commercial.
That's great. This webinar is a high-level understanding and is geared towards academic research environments and startup companies in industry. So that's a great mix of people for the audience, as well as those individuals responsible for keeping the employees and students and postdocs in those environments safe on the IH side of things. So it's a great mix for this, and this should be a useful overview of a lot of the issues and concerns that you may or may not know regarding these types of materials. So to get things started, Mark and I have worked together for a number of years now, and we decided to put on this webinar in large part because of the way that these materials are perceived is not necessarily the best way to go about thinking about how these materials affect the people that are making them. For example, in the biomedical engineering field, which is where my expertise is, when you look at all of these people trying to cure cancer, for example, using these types of nanotechnologies, you want them to be successful in curing cancer, but you don't want them to give it to themselves in the process. So Mark and I basically use that tenet. It can go across all of the different industries that utilize these materials, but that's the basic issue we're trying to address, is the ability to work with these materials in a safe manner so that people can change the world without causing harm to themselves in the process. So with that, we'll go into an overview of nanoparticles. So the ASTM Committee on Nanotechnology defines nanoparticles as substances with lengths in 2 or 3 dimensions between 1 and 100 nanometers. So that could be a spherical nanoparticle, or it could be a carbon nanotube, a silicon nanowire, these types of materials. These, obviously, looking at the ones I just mentioned, can have a number of different shapes or geometries, including spheres, rods, discs. They could be amorphous, they could be agglomerated, and they can have properties that differ very widely from their parent compounds that are on the micron or millimeter scale. And these types of materials are usually found in the laboratory in the form of a powder or a suspension or some sort of matrix environment. The types of industrial applications that these nanotechnologies have are seen widely in biotech and pharma, as well as cosmetics, diagnostics, and sensors, electronics, pigments, ceramics, textiles, and even water purification. And that's, a lot of these are owing to the properties that these types of materials have. To support those industrial applications, the academic side has a number of different areas of research where these nanomaterials are being engineered, that being chemistry, physics, biology, different engineering fields, and a lot of medical applications, whether that's diagnostic radiology or these types of disease state treatments in biomedical engineering. So they're widely spread, and one of the things that we're interested in getting the point across for is a lot of these industries are not necessarily chemistry or material science focused. So the training that exists for working and handling these types of materials might not be ideally suited for keeping the people safe that are working with developing these types of materials. In traditional lab settings, as I mentioned earlier, these are usually in the form of powders or dispersions. They can be agglomerated, so they can actually be present in more micron-sized agglomerates, and this is important when Mark goes through some of the PPE that's needed to work with these types of nanomaterials. Those agglomerates can be dispersed by a number of mechanical energies that you could have in process. They can also be dispersed upon interaction with your body. And those resulting dispersions or suspensions could be present in aqueous or organic based solvents. Some of the properties of nanoparticles that make them traditionally different than A, their parent compounds, or B, most other materials that you might experience on a more macro scale. They have a high surface area to volume ratio, and in general a high surface area. That's what makes them very good at things like water purification. Nanotubes, nanowires, these and rods could have very different aspect ratios than something that's a spherical particle, which has implications in clearance routes through different exposure routes, whether that's inhalation. The aspect ratio actually controls part of the feature that interacts with your tissues. Another thing that interacts with tissues predominantly is surface charge. Obviously something that is positively charged interacting with your cell membrane is going to be very different than something that's negative. So the sizes, obviously very small, capable of being dispersed airborne and following Brownian motion to make its way into your body through an inhalation and respiratory route. The size and shape and geometry of these is a precursor for understanding how they are going to interact with your immune system once they have penetrated your physiological spaces. They can have different porosities. They can be biodegradable or not. And they can have surface functionality that causes the ability to change not only surface charge but surface wetting capabilities, which is a unique characteristic that spans most of the industrial applications that we mentioned earlier. People can also do further chemistry to attach things to the surface of these, and biomedical engineering, the ability to encapsulate active pharmaceutical agents. So there's different things that you're wanting to be able to characterize initially, so that when you make a new material, you're even making a material over and over again, that you know how to characterize those, so you can start to think how these materials are going to interact with your tissues. So now we're going to go into the different classifications and types of nanoparticles. There's basically three classes of nanoparticles: solid, vesicular, and macromolecular. The solid class is usually natural or synthetic polymeric nanoparticles, metal nanoparticles, whether those are noble metals or metal oxides, carbon nanoparticles, whether those are nanotubes, carbon blacks, or smaller synthetic carbon molecules like fullerenes, and ceramic nanoparticles, your silicon based particles, titanium, aluminum, all have very different utilities in these different industrial applications. Vesicular, where you're talking about micelles, those are widely seen in solubilizing agents, liposomes and polymerosomes, and these lipid stabilized particles are more in the biological and biomedical engineering side of research for a lot of different applications, not just drug delivery. And macromolecular nanoparticles, these are things like dendritic polymers or chelating molecules that are used in a variety of different aspects that are owing to their incredibly high surface functionality in a very small surface area to volume ratio. So very easy to target and change the surface character to display ligands that you're interested in. To go a little bit more in detail, so polymeric particles and these next series of slides will look at questions that you want to ask yourself as a researcher or an EHS staff member when you're doing an inspection of a lab. You know, what are the people actually working with, what are they doing? So polymeric nanoparticles can either be biodegradable or not. Biodegradable could mean it's degrades into smaller monomeric subunits by interaction with water or enzymes when they're come in contact with your tissues. Non-biodegradable, very different, and they're going to be very different classes in terms of how your body is trying to clear these materials through the reticuloendothelial system. Metallic nanoparticles, something like a gold nanoparticle versus a quantum dot. You have a noble metal which has minimum toxic capabilities, or a quantum dot which has, you know, cadmium selenide, for example, that is, you know, highly toxic heavy metals. So these different metals, to know what they are, to know if they're an oxide, if they're a core shell type of particle where you're either coating these particles with different types of materials to change the surface characteristics. But a lot of these metal particles span the biomedical engineering side, so your iron oxide and gold nanoparticles are used predominantly in imaging techniques for MRI and CT. Quantum dots are actually used in producing LEDs for televisions in a lot of interesting applications. Ceramic nanoparticles, these are intrinsically either porous or not porous, and they can take a variety of different geometries that can make them very interesting to interface with your immune system cells. These have wide uses in a variety of different industries, from anything from textiles to solar energy. Carbon-based nanoparticles, these have interesting properties, very high surface area, and are used widely in filtration systems. Carbon nanotubes are these different structures that have very high aspect ratios, and carbon nanotubes are where a lot of the EHS concerns had started when NIOSH recommended exposure limits for these, because owing to their surface characteristics and aspect ratio, they behave very similar upon inhalation to asbestos type particles. So obviously cancer-causing mesothelioma. So there's restrictions initially placed on these. Carbon black, which is predominantly used in pigments for printing. That is a class of materials that not only could have potential issues from a foreign body response standpoint, but also in the right situations is a combustible dust. So a lot of different EHS problems, so not just what is it doing to the employee that's being exposed, but what is it doing to the environment they're working in. Vesicular and micellar based nanoparticles, these are predominantly used in biological applications where you're looking to mimic cell membrane architecture. And that's one thing to think about, is a lot of people that are engineering these materials, we're designing them to affect the body, usually in a therapeutic manner, but you know, you're still designing them to penetrate your defense mechanisms. So these are a variety of different cell mimics for different applications. The macromolecular nanoparticles, dendrimers, dendrons, these hyperbranched polymers that have a huge surface functionality, great for chemists and chemical engineers to manipulate these to make them have properties that are suited for a variety of different applications. Macrocyclic chelators, for example, gadolinium DTPA, which is something that everybody gets if you go in for a T1 weighted MRI session. But these can also have chelated to them radionuclides and other heavy metals. So they're one of the things to bring out is the fundamental nanoparticle might not have a toxic concern, but what it's carrying or what it's coupled to could actually have some pretty deleterious side effects associated with it in terms of tissue interactions. And then going into what everyone likes to do with these systems is put as many things on the surface and as many things inside of them for all of these different biomedical engineering applications. So again, here, a lot of these things could be benign, but taken as a aggregate and in combination with each other could actually pose some issues for occupational health issues and concerns. So, things now to ask when you're looking at a lab setting, how are these nanoparticles being prepared, handled, and disposed of? So to be armed with all of the different methods for producing them, whether they're precipitations, emulsifications, synthetic methods, self-assemblies, catalytic growth, chemical vapor deposition, or something as simple as taking a larger material and milling it or grinding it down into a nanopowder. Are those materials then functionalized so that their surfaces are inherently different than the starting material? What does that create in your lab environment? And are those materials encapsulating something else that's potentially hazardous to health and environment? As far as characterizing them, you want to be able to know what these things look like and how they behave in different, whether they're dry powders or dispersions. So the characterization methods are something that are of importance to most labs. Things like electron microscopy, light scattering techniques, size exclusion techniques, laser scattering, laser diffraction techniques, to sort of figure out what you're looking at from a size perspective, as well as a shape and geometry and surface architecture. You want to know what those characteristics are so you can start to think about how your body is going to respond to them invading that space, whether or not they're charged particles, hydrophilic, hydrophobic, and what's giving rise to those issues, because the main thing when your body sees one of these types of materials is it's going to have a protein adsorption phenomenon that's going to cause complement cascade and further foreign body response to try to clear the system. And how are these different characteristics going to change the extent to which that happens? And then looking at how these particles are handled and different materials are handled. Some of these procedures are being done in a clean room. Some of them are being done in an academic research lab. And are they handled as a powder? are they handled as a dispersion, and what engineering controls are actually in place and available to enable safe handling and disposal of these types of materials. Something that most people know about these is, given their high surface area and charge, in very dry environments, these materials are very difficult to weigh out, for example, or aliquot safely. They're very easily to become airborne and succumb to Brownian motion to invade your respiratory tract. So, there are measures to take with engineering controls, but something as simple as breaking the electric field with around a little tube that you're weighing these particles out of is something that you can accomplish with a nitrile glove and a certain technique to establish an electrostatic discharge based scenario. And these are things that are tricks of the trade. But with that, I'll turn it over to Mark, and Mark will go through the industrial hygiene side and what we're trying to do to help people work with these materials safely.
Thanks, Jason. Thanks, Jason. Talking about the difficulty in weighing out dry powders, Jason was telling me a story of what was it? A lab assistant who was trying to weigh out some material.
Yeah.
And I'll let you say it because you can do it better than I.
We had roughly 10 grams of a biodegradable nanoparticle, and this postdoc was weighing them out for the, not the first time, but definitely not an expert in doing these types of things. And he came back to me with aliquots of 50 milligrams, but he only had 2, and the other, basically 9 grams of material somehow went into the air. And these are things that happen, and so what do you do to protect yourself and those around you and the environment from these types of mishaps?
Yep. Those electrostatic charges, they'll get you every time. Hey, and James has put up a question. Has your facility ever conducted a nano assessment? And what Jason and I were just curious as to how many folks have conducted a formal nano assessment. Maybe you're doing it for control banding purposes. Maybe you're doing it for a PPE hazard assessment. But we're just curious as to how many folks have done that.
Thanks for teeing that up, Mark. We'll give everyone a few more seconds, and I'll share the results again.
Okay.
Okay, looks like we got just about everyone. We'll push those out. So Mark, 50% have never conducted a nano assessment, 30% are unsure, 13% have done it once, and 8% have done it several times.
Okay. Well, thanks, James. I think it's important that everyone who has people who are working with nanomaterials get into the mode of understanding what's going on and conducting those hazard assessments. And we've got some tools and comments that we'll talk about. One thing, when you're dealing with nanotechnology, there's a lot of science involved. We've been working with nanomaterials for a long time. Actually, people have been exposed to nanomaterials since the first person created fire. Sea salt from mist evaporating over the ocean generates nanoparticles. Soot from fires, tiny grains of sand from storms. These are all nanoparticles. Our bodies have been exposed to them for a long time, and we've evolved so that we can handle those types of nanomaterials fairly well. But now we're getting into the situation where we're engineering them, we're changing the properties, and as Jason said, we're vastly increasing the surface area and charges of these materials, so they no longer act like their parent materials. And we can expose ourselves to nanomaterials whenever we're making it, manufacturing it, transferring it, weighing it, handling it, cleaning equipment and glassware that's been in contact with it. So we just have a large range of exposures. We have studied, or there have been a lot of studies done on the carbon nanotubes, and the picture you see here is a multi-walled carbon nanotube penetrating the pleura of the lung. People love to flash these photos around, but it looks awfully much, like a long fibrous particle. And some research has shown that lung cancers or mesotheliomas have been produced in mice by multi-walled carbon nanotubes. So that gives us some concern. So as we said, the molecular structure of nanoparticles and that great surface area gives them very different chemical properties than the parent compounds, and it's a real challenge from an occupational health perspective because we've got a couple of things. We've got very limited toxicological data. A lot of these materials haven't been around for a long time, and people are constantly making new ones, and we don't really have good exposure data or exposure information for these materials. They tend to be very light, so measuring just by mass is not a good way to do it. And we do have a lot of folks, there's a lot of research being done by NIOSH, NIH, EPA on these materials, and they all are urging people to be very cautious with our exposures to these materials because we just don't know what the long-term effects 20 or 30 years down the road may. And then there's the issue of combining FDA-approved materials with nanoparticles. I'll throw it to you, Jason. If I'm using an FDA-approved material, isn't that a good thing?
Not necessarily. So it's a misnomer that FDA-approved materials in the world of nanotechnology means safe. So what people are doing with these materials, even if they're benign in terms of the FDA's regulatory pathways, what we're engineering these to do might not actually be as safe as the parent compound that the FDA has approved. So it's a bit of a misnomer that you walk into a lab and they say, oh, I'm using FDA-approved materials, I'm safe. That's not necessarily the case.
Okay, there goes that excuse. Occupational standards for nanomaterials are emerging. We have some for titanium dioxide and ultrafine titanium dioxide. We have a limit for carbon nanotubes and carbon fibers. It's based on basically limited detection, and it's a mass-based standard. And there's a draft current intelligence bulletin out for silver nanomaterials that will drop it to 10 micrograms per cubic meter. A number of manufacturers have established occupational exposure levels or limits for nanomaterials that they make or use internally, and a number of folks are using controlled banding systems to try to get a range for the level of control you need for different types of exposures. Interesting that NIOSH chose a mass-based recommended exposure limit over counting of the fibers for carbon nanotubes, but it appears that they went the mass-based route because animal toxicological studies are mass-based, and counting protocols haven't really been developed, although people are working on that for how we'd count the fibers. The other issue is clumping and agglomeration. When these materials are airborne, they tend not to sit there as single fibers or single tubes. They lump and clump together, and it's once they're in your body, in the fluids of your body, that they'll then basically separate and cause issues. So a lot of technical issues around how to come up with a good means of evaluating exposure. Common processes, and we'll see these in both a lab base and in a small scale manufacturing or even a larger scale manufacturing, is the harvesting of the nanomaterials, regardless of how they're made. they're typically going to be in some sort of a reactor, and we'll have to open it up, reach inside, use some tools to scrape out the deposited nanomaterials, and that's a source of exposure. We can enclose the reactors like this photo here. We can attach basically gloves, make it a big glove box, but you're still going to have to reach in, service the equipment. Then there's, what do you do with the material once you've gotten it made? You've harvested it, it's not in the reactor anymore. Now you need to bag it and package it. Downstream, the folks who are going to be using these materials are going to mix them, weigh them, sonicate them, do what they're going to do with that. And then we have the whole issue of, okay, the equipment, whether it's small or large, you're going to need to maintain it. Typically you'll have local exhaust ventilation systems that are servicing this equipment. You'll need to be doing filter change-outs. You'll have accumulations in the ductwork. So all of that needs to go into your risk assessment for working with your nanomaterials. And as we said, we just don't have a lot of data for the exposures to people, doing especially lab-based work. And some research has shown that the handling of dry powders, again because of their electrostatic properties and their very tiny size, even inside a fume hood, can result in a significant release of airborne particles into the lab environment. And once they're in the lab environment, they tend to linger, for some time. So they'll be present in the breathing, potentially present in the breathing zones of the workers in the lab. These photos here, just some, the upper left, this is a harvesting operation. Basically, you see we have a chamber. The way we make the nanoparticles is we essentially vaporize them. They condense out. This is a real oversimplification, but then they'll condense out inside the chamber. We need to scrape them off to get them. Then we need to clean it, the center photo. We need to handle those materials once we've gotten them out. We need to weigh them. And then downstream, we're going to be taking the produced nanomaterials and putting it in other systems. We could be making, say, ink for inkjet printers. There's a big use of nanomaterials for that. So it goes from the lab to the industrial operation, and they all have the potential for employee exposure to that. Exposure pathways, you all remember from your Toxicology 101, inhalation is our primary route of exposure, and airborne nanoparticles will enter the lung, and they can penetrate the lining of the trachea and the bronchioles. And remember that, especially in the biomedical setting, these particles that we're working with have been engineered to fool and defeat your body's defense system. So once they get into you, they're going to move around. They may even go to other organs or pass through the blood-brain barrier. So a lot of potential for exposures that we really don't contemplate for the parent materials. Some cases, nanoparticles have been shown to migrate through the skin and be circulated in the body, and this can be of concern if the particle's allergenic or carcinogenic, so even tiny quantities could be significant. Ingestion, as with any material, if we're not following good hygiene practices, we have plenty of potential for contamination of your hands, your food, your face. And Jason and I have been in a few labs where we would say that ingestion is definitely a potential for working with nanomaterials. Just contaminated tools, equipment, benches, poor use of PPEs. So it does happen. And then we always have the injection route of exposure, typically accidental. A little bit on some thoughts and issues around evaluation and control. Obviously, we want to control our exposures to nanoparticles, and the emphasis right now is primarily on the control aspect. We don't have a lot of exposure limits or even methods to analyze for exposure, but we do need the data. We need to know what those exposures are. So we'll talk a little bit about some of the tools we have for that. the point we talked about earlier is exposure to nanomaterials in the air is going to be in the form of aggregates and agglomerates. You know, we've made the material, it's all lumped and clumped together, it's in the air, so it's not a free-floating particle in most cases. So the size of these particles that we're measuring by impactors or mobility analyzers doesn't tell us the state of agglomeration. It doesn't tell us anything other than we've got this mass of material in the air. We don't know how many thousands or millions of particles that we have. And this is a electron micrograph of an agglomeration of carbon nanotubes. You can see all the little individual tubes. So imagine, you know, these floating particles, which are agglomerations of nanotubes. This gets into your lung. Now in the fluids of your lung, they're all going to separate and they're going to go their merry way. And you know, that's the type of exposure we're looking to get some assessment on. So evaluation strategies are being developed by a number of researchers. I'm really in favor of the non-mass based metrics such as particle surface area or particle number, a lot like what we do to count asbestos particles. And way back in the early days, asbestos was a mass concentration, not a particle concentration. So it's tracking, you know, a similar way. Evaluation should consider, okay, what are our sources of emission? You know, is it a harvesting operation? Is it a bag dumping operation? Because we're going to be typically looking at mass-based concentrations, okay, I need to know what my background concentrations in the air are, what's my area monitoring showing? Am I getting dust? Am I getting diesel emissions? Am I getting other things that can confound my measurements? I need to know what those are. It's very, very useful to determine air concentration by direct reading instruments. A lot of these operations are very brief and sudden. I spoon something out, I'm going to get a very brief high concentration of something when it gets dispersed. So I'd like to know what that looks like. Oops. So to assess our occupational exposure, again, we want to know the background. We really want to know the particle size distribution. You know, what's the size range of these particles? That's going to tell us, you know, where in the body it's going to go. Is it going to be thoracic? Is it going to be deep lung? You know, how am I going to do that? And one of the things folks found out early on is that our conventional IH pump-based filter sampling really isn't the best solution for exposure assessment. It just doesn't have the sensitivity. You can't get down to those nanogram levels of materials. So mass concentration isn't well suited to toxicity assessment. However, you know, what else are we going to work with? We need to get data from something. So we're a bit of a dilemma. None of the existing instruments used for monitoring really give us specific information about particle concentrations at the very, very, very small end of the size range. Tools we have: cascade impactors. Hard to find an industrial hygienist who hasn't spent a fair amount of time working with these. And these are good, you know, down to about 250 nanometers. We collect it in stages so we can get a good idea of the aerosol mass distribution. Tapered element oscillating microbalance, used heavily by the EPA, measures aerosol mass concentrations across a wide range, micrograms to grams, and particles are collected on a glass tube mounted filter, and as you change the vibration frequency, you collect different mass concentrations. A scanning mobility particle sizer has a size range of 2 nanometers to a micron. This creates an electrical field, and the particles drift according to their electrical mobility. So this is a nice way to get a sense of the electrical mobility of a particle. Downside with this is that it uses a radioactive source, so lugging this thing around might cause some issues. An electrical low pressure impactor basically charges the particles by corona effect at the inlet to the sampler, and then they're going to deposit themselves at the collection stage that corresponds to their aerodynamic diameter. A condensation nuclei counter. These are the concept has been around for a long time. Basically, we have a chamber saturated with an alcohol or water vapor. The tiny particles will condense, and then the particle growth will enable the detection of the nanoparticles. And we can get down to about 3 nanometers in diameter with some of these instruments. A little bit heavy to lug around, though. And this is an instrument that measures the surface concentration of an aerosol that would be deposited in the tracheobronchial or alveolar section of the airway. And again, we're charging particles at the surface, and the number of charges carried by the particle is related to its surface area. So with this, we're going to be able to find out, again, the charge of the particle. Lot to go with out there. And then we didn't even talk about electron microscopy that is getting very, very popular. So I've got nanoparticles. I'm working with them. What do I do? Well, basically, and this is what most folks are doing, working with engineered nanoparticles, you want to handle it the same way as you would if you're working with any chemical or research chemical of unknown toxicity. A lot of similarities between working with potent compounds in a pharmaceutical setting as working with engineered nanoparticles in an engineering setting. Same principles apply. So if you're handling materials outside of a fume hood, biosafety cabinet, or a glove box, I think we all know the drill, right? Latex or nitrile gloves, although most people have X'd latex out of their systems because of the allergy concerns. Double glove, always do glove changes, change the outer glove in your containment, in your fume hood or biosafety cabinet. You want to prevent particles from becoming airborne. Wear a splash goggle if you have nanomaterials in suspension. Lab coats should be regularly laundered. You want dedicated lab coats for the lab you're working in. And then respiratory protection. A good particulate filter whenever you're working with nanoparticles and there's a potential for their release in the air. See what the safety data sheet says about nanoscale materials that you're purchasing. You're going to find that because there's not much data, many SDSs will basically give you information on the bulk parent material, so it may not be of much use to you. Once you get your nanoparticles into solution, you can handle them on the open bench typically. And transporting nanomaterials, you always want a sealed secondary containment device, and clean those work surfaces. Wipe them down regularly. Even if you're working with nanoparticles in solution, that solution dries out. Now you have a dry nanoparticle on your surfaces, and contamination may not be visible to the naked eye. We're dealing with very small amounts of materials. Clean up all spills immediately. Dissolve your waste material in an appropriate aqueous or non-aqueous solution. Manage the waste as the parent material. Again, if you're doing a cleanup, take your time, make a safety plan or a health and safety plan first, figure out who's doing what, make sure you got the right PPE, double nitrile gloves, probably respiratory protection. Always use a HEPA filtered vacuum, or get the material wet or into solution before you start handling it. Wear an appropriate respirator and never brush or sweep or blow with an air gun any kind of a dried nanomaterial. What we've seen with research nano safety programs, they vary greatly. We've seen the gamut from no program to a one-page handout to a many-page, very detailed SOP. And it just depends on the maturity of the organization and how much work they've been doing with nanoparticles get those safety programs in place. And then medical surveillance should always be a part of any research nano safety program. Common planned elements, identification and notification or registration of the EHS group, so people know, who's got what. Engineering controls, chemical fume hood, biosafety cabinet, laminar flow hoods, ESD or electrostatic dissipation controls. You again, depending on what you're working with, you might find that this could be very useful. SOPs based on a risk assessment of your materials. Follow the ALARA principle, the as low as reasonably achievable principle. Again, we're working with materials. We don't know what the long-term effect may be. Appropriate PPE, we've talked about that. Make sure you have the right emergency and waste management procedures in place. And the photo here is someone just doing a boron nanotube harvest. So you can see the PPE that this person is wearing. We always want to follow our hierarchy of controls. Again, it's just safety 101. Select the highest level that's feasible, eliminate, engineer it out, administrative or work practice controls, and then PPE is our last resort. And then I thought it would be useful to show you guys, if you haven't used it, it's worth a look. Lawrence Livermore Labs a number of years ago came out with a control banding nano tool. They're up to version 2.0, and to use it, you download it for free. It's a simple little utility, and you type in information about the nano materials that you're working with, and some of the info you type in is surface reactivity, particle shape, particle diameter, solubility, carcinogenicity, reproductive toxicity of the parent compound, et cetera, et cetera. And then for probability, the dustiness, mistiness, they have a way to calculate that, number of people with similar exposure, frequency of operation, a number of parameters, and out of that you can get a risk level 1, 2, 3, or 4. So risk level 1, general lab, on the open bench top. Risk level 2, fume hoods or local exhaust. Risk level 3, you want total containment of the system. And then risk level 4, you really want to, okay, I need some real help here. Either better evaluate my toxicity, better evaluate my controls, get some people who have worked with similar operations. So I'd encourage you to take a look at that, because it's simple, but it's a good tool. So based on that, I think we're kind of sort of on time. So this is where James, I think, asks if folks have questions.
Thank you, Mark. And we will, at this point, open the floor to questions. Got about 5 minutes. I did receive some questions throughout the webinar, and I think we'll do a quick lightning round. I do want to remind everybody that you will receive a copy of the presentation. Got a couple questions about that, and you'll see that in an email tomorrow morning. So we'll go on to Mark, let's go to that last slide. I know we have your contact information that we could share. There we go. And so feel free to email Mark or Jason directly if you have follow-up questions. You can also give us a call at Triumvirate. And there's also a link at the top. So as a thank you for attending, we're offering a free consultation, just a chance to speak with a professional like Mark and Jason about your current strategy for managing nanomaterial exposure risks. And they'll offer some recommendations for improvement. So I'm going to shoot that over in the chat box if you're interested in taking a look at that. So we got some questions early on. Let's get to those. I think, Jason, we'll start with you. This is coming in from Paul. Have the hazards for drug delivery via nanoparticles been officially assessed? How can the variability of drugs affect the stability of nanoparticles?
So the short answer to that is no. These types of systems, one of the reasons that field exists is to bring these types of technologies for clinical utility. So all of the research that you're doing to advance one of those products from an idea to a clinical product is looking at, through the FDA's regulatory guidelines, what is the toxic concerns and what is the safety of the product, and the research programs are designed to answer those questions. So a lot of what people are working with in a research setting was something they've invented or is brand new. The characterization of the safety and toxicology of those compounds is not well understood. While there are trends in looking at these types of drug delivery systems, interactions with the immune system and tissue interactions, those are still research topics that are not well understood even by the regulatory agencies. And that's something that I came across when bringing a product from conception to discussions with the FDA. And it's just something that people need to understand, that they don't know a lot about it and how to go about trying to answer those types of questions.
We'll stay with you, Jason. This one came in pretty early. Fred wants to know, when you say biodegradable, does that also mean compostable?
In certain applications, yes. These materials that degrade by interaction with water would have both of those properties. But biodegradable in these settings could also have the ability to degrade by interactions with enzymes or other mechanisms that are chemically tied to and biologically tied to the materials. So in certain cases, biodegradable can mean compostable as well.
Thank you, Jason. Mark, let's shoot this one over to you. Is an N95 respirator effective against nanoparticles? In other words, is that enough?
Yeah, either N95 or an NRP 100 respirator would be effective. A lot of people are concerned that they see the N95 rating and it's at 0.3 microns, and they say, oh, it's only 95. It's only 95% effective at 0.3, and then that, if it gets smaller than that, it's going to be, they're all going to get through. But the 0.3 microns is just a notch in a filter efficiency curve. It's where you're changing basically the mechanisms of deposition on the filter. The particles, when they're big chunks, will hit the filter fibers or get stuck between them, and as they get smaller and smaller and smaller, then it's electrostatic forces and Brownian motion that makes them basically float very slowly in the airstream and stick to fibers. So a filter is least efficient at that 0.3 micron, and it gets actually much more efficient as we go smaller. So the short answer is yes, and the long answer would be a whole other webinar on how filters work.
We'll take care of that next time. Mark, let's follow up here. Kim wants to know, are there special handling suggestions for respirator cartridges after using, storing for reuse, and disposal?
Typically not. I mean, what we're working, if the question is, are they hazardous for disposal, I would say typically not. You're not going to get much, if anything, in there. Go buy the parent material. Try to figure out what's on it. But all the cases I know of respirator cartridges, basically you wipe them down before you take them off, like you do your PPE, and then you can dispose of them normally.
Thank you. And let's end on this question. I know we have several more, so I'll definitely get Jason and Mark to help out in following up. But both of you, how do you recommend EHS personnel garner commitment from researchers and administrators to report nanoparticle use?
That's the big question.
So I think, and this goes to why that first poll question was asked, is this really needs to start in the academic environments where people are actually being trained on utilizing these. and startups and the bigger industries, the thing that is the hardest to do is to get people around their egos, and the ability to have people know that there are things that are unknown about these types of materials. So it's more of a shock and awe type of approach with people that think that they've been trained properly. But the fundamental, if you look at the programs where they were trained, some of them may or may not be suited to actually perform those duties in a safe manner. So we're looking, to basically have people that think, and this is traditional of chemists. Chemists think that they know everything that they make and what it can do, and how to protect themselves. And a lot of synthetic chemists, will think they're invincible and don't need to wear gloves and eye protection, and because they know that the compound they're working with is capable of this. With nanoparticles, not only are the people working with them not necessarily material scientists and chemists, but even if they were, you'd want them to know that these materials, there's a lot of unknowns, and caution is required even if you think you're invincible. Anything, Mark?
No, education. They need to be educated as to what these are and what the unknowns are and what the potential downstream liabilities are.
Well, thank you. I think we'll wrap it up there. Thank you, Jason and Mark, for sharing your knowledge with us today. I want to thank our audience for attending. And again, please be on the lookout tomorrow for a copy of the deck, as well as a survey. We'd love to hear what you thought. Any ideas for future webinar topics or improvement are appreciated. Thanks again, everyone. And thank you, Mark and Jason. Have a great rest of the day.
Yep. Thanks, everybody.
Thanks.